All right, so we are recording now. All right, so first, a brief overview of the brain and how the brain works. All right, so here we have two pictures of the brain. We have on the left a picture of the brain from the side, and on the right we have a picture of the brain looking down on the brain. And the brain has this external covering, but it also has internal structures. Those internal structures tend to be more primitive areas and do what we kind of consider less complicated, although certainly they're complicated tasks, more subconscious related processing. And parts of the brain that we'll talk extensively about today that are internal parts of the brain are the basal ganglia, which does a lot of sort of regulatory control of brain activity, and a particular area of internal in the brain called the amygdala, which is involved with the value that we place on external stimuli or internal stimuli, but the value we place on stimuli, the emotional value that we place on stimuli, and the nucleus of accumbens, which is a center for our reward, our pleasure center. And then this outer part of the brain we call the cortex, this sort of wrinkly area, and that is where we have sort of our most complex cognition. It's where we have all of our sensory perception. It's where we have all of our motor system control. And in particular, the frontal lobe that we'll spend a lot of time talking about today is where we have our sort of most complex thoughts, executive function that Penn mentioned in his song. We'll talk a lot about that. And it's where we store our memories down in the temporal lobe. And then we also have this area back here called the cerebellum that we know is important for coordinating sensory and motor movements, but that was sort of its original function that was originally discovered. But now we've discovered that it does a lot more nuanced cognition types of things. It's involved with helping us with timing of different things, and it appears also to be involved in ADHD. All right, we have two hemispheres. We have a left hemisphere and a right hemisphere. They're connected. We're not really going to talk a whole lot about the two different hemispheres because the frontal lobes kind of act as one unit. They don't really, left and right don't really do a lot of different things. All right. I saw a note in the chat. Can everybody see the slide okay? Did we get that worked out? I'm gonna make sure I'm sharing it right. I can share the PowerPoint if that makes it easier. Is anyone still having a problem? Raise your hand if you are. Nope. Okay, everybody's good, looks like. All right, so we'll just keep it like this then. Okay, so in general, the cortex, we can talk about it being organized into 4 different lobes. We have the occipital lobe back here in the rear part of the brain, and it almost exclusively does visual processing. We have our parietal lobe, which is just in front and to the top of the occipital lobe. And the very front part of that, in the darker red there, is our center for touch processing, our touch sensations. The rest of it is kind of complex visual processing. And we have our temporal lobe down here in orange. That has our center for hearing in the dark orange. It's also where we have language comprehension, and it's primarily involved with storing long-term memories and accessing those memories. And deep inside the temporal lobe is where we find those internal structures, the amygdala, and the hippocampus, which are involved with memory and emotional value with stimuli. And then finally, we have the frontal lobe. And the frontal lobe is where we're going to spend most of our time today, along with some of the internal structures like basal ganglia and the nucleus of accumbens that I mentioned. So there's a lot going on in the frontal lobe, and it only is about a third of our overall brain. yet we have this dark purple strip here, which is our motor cortex. We have our center for our smell perception is there. And a lot of this area right in front of the primary motor cortex is involved with motor processing and sequencing and motor planning and initiating movements. This particular area right here is Broca's area that's involved with language production. So really we only have about half of the frontal lobe that's available for this stuff that we're going to talk about called executive function today. And that's where we have things like short-term memory, complex thought, reasoning, emotion, personality, things like response inhibition, concentration, all of those things that we sort of think about as our highest sort of thought processes. So really pretty small amount of the brain that's responsible for doing a lot of that work. Now, with that said, that small part of the brain is widely connected to a lot of other parts of the brain to sort of share that processing power and to do those complex things. All right, just a little bit more background, and then we'll get into development of the brain. As I mentioned, I think I sort of went through this on the previous slide, we have these specialized areas for different perceptions that are localized in different places in the brain. And in general, our brain is what we call contralateral to our body. So the left hemisphere of our brain is receiving inputs, sensory inputs from the right side of our body and giving motor outputs to the right side of our body, and vice versa. The right brain is in charge of the left side of our body. Like I said, with the frontal lobes, we're less concerned about that. It's less lateralized and they act more like a whole unit there. Language and speech are in these special areas, again, taking up some more space in those frontal lobes. All right, so moving from sort of an overview of what these different brain structures do and where they're located, I want to just sort of give a review of neural signaling and neurons and some of the important parts of those. All right, so here we have a series of neurons that are connected to one another. And so we have an output from this neuron at the top that's giving chemical inputs to this next neuron. That neuron is going to send a signal then to the next neuron. And this is the way that our brain communicates. So when we talk about the frontal lobe communicating with other parts of the brain, this is the process by which that happens. So within a neuron, after it receives those neurotransmitters get converted into electrical signals. And those electrical signals travel as inputs through the things that we call the dendrites here that are receiving the inputs. And they add up in the soma, the cell body. And if there's enough inputs, then the neuron decides that it's going to send an output. And in that case, the output travels through the axon, and we call that the action potential. And we can think about the axon essentially as the long wire. The soma and the dendrites are sort of the decision makers. Do I have enough input coming in? Is it proper for me to fire an output? Once that occurs, the action potential travels down to the end where we have these things called axon terminals. And that's where we're going to now convert this electrical signal that happens within a neuron. It's very fast and can travel long distances back into a chemical signal. And so why do we have these two different types of signals? Well, the electrical signals can travel very quickly over long distances. So if everybody wiggles their big toe right now, you just used two neurons, one neuron that went from your brainstem all the way down your spinal cord, about 3 feet in most people, or 2.5 feet, 3 feet, to another neuron in the bottom of your spinal cord that then traveled all the way down your leg, another 3 feet, to cause those muscles to move in your big toe. So with just two neurons, we can travel that 6 foot distance from, the top of our the base of our brain all the way down to our big toe, and we can do it very fast. Right? Now, we don't have electrical signals typically between neurons. We convert those signals back into chemical signals that are going to float across that really small gap in between neurons that Ramon E. Kayel discovered, called the synapse. And this is a short distance, so it slows down the transmission speed a little bit, but not really. It's not a huge delay in that signaling. And the reason that we want to go from an electrical signal into a chemical signal is that because those chemicals have specific shapes, we can activate different receptors on the other side, and those different receptors can do different things. So we can have one chemical signal that activates a receptor that turns that next neuron on, and we can have another chemical signal that acts with a different receptor, for instance, to turn that neuron off. And so this selectivity of the chemical shape of the receptors and the transmitters that fit in them is the benefit of giving up that little bit of speed in order to get that selectivity of what we're able to do, the types of we can give different signals to the next neuron. Electricity is just on or off. You can't control, you can't have selectivity with that. You just get a binary sort of on or off type signal. All right, so why do you care about this? Well, you care about this because we're going to talk about plasticity today, development, and we're going to talk about drug effects. And drugs have their effects by acting like those neurotransmitters, or by increasing or decreasing those neurotransmitters to influence the neural signaling in the brain, right? So it's really important that we understand how this neural signaling works and how these receptors and chemicals work in order to start to understand drug effects. And the more activity that we have with plasticity, the stronger connections become. If we don't use connections for long periods of time, they can weaken over time. And all of that has to do with the chemical signaling and electrical signaling that are happening there. All right, so as I mentioned, neurotransmitters have this sort of specific shape, chemical shape, and they fit in particular receptors. So here we see two different chemical shapes, and each of them only fit in the receptor that matches their chemical shape. Again, here on the side, we see a sort of more complex, nuanced version of that, where we can have one chemical that may fit in multiple receptors, but not fit in others. And so later on, when we talk about dopamine and we talk about norepinephrine, we're going to talk about multiple receptors. So there's at least 5 different dopamine receptors. And what that means is there's at least 5 of these different types of receptors that do different types of things in the brain that dopamine can all fit in. Right? So dopamine can activate all of those, but norepinephrine, another neurotransmitter, for instance, can't. So it's going to come in and activate a different set of receptors because of its shape. Right? One of the reasons we care about these different receptors is even though dopamine can activate all 5 dopamine receptors, we can create synthetic chemicals that only activate 1 or 2 of those dopamine subtype receptors. So they're not able, unlike dopamine, they're not able to activate all the different receptor types. So that gives us some more selectivity with drugs sometimes than neurotransmitters have. So why is our brain set up this way? Well, for 2 reasons. One, there are no walls in the brain. There's no physical barriers. And so if we have 2 areas that are next to each other in the brain, they want to use different neurotransmitter signals so that they don't interfere with each other, right? So that there's not what we call crosstalk, because if some dopamine neurotransmitters that are being used in the reward center float over to this other area next door, it's not going to activate that area because that area is using a different neurotransmitter in its signaling pathways. Right? So that's one reason. The other reason is, as I mentioned, we can have different receptors. Those 5 different dopamine receptors all do different things. Some excite the neuron, some inhibit the neuron. And so we can do different things with those different receptors. So in general, some neurotransmitters that you might hear mentioned today that we want to sort of go over. We have 2 neurotransmitters that are generally used with what we call discrete signaling. So these really are kind of like on or off signals. We have glutamate, which is sort of a general excitatory neurotransmitter. So that's, if one neuron wants to turn another neuron on, it's going to use glutamate. If we're trying to communicate between areas, the glutamate and the GABA being the inhibitory neurotransmitter, are really the way that areas turn each other on or off and create those signaling pathways. So they're kind of like the workhorse of the central nervous system in terms of controlling signaling. Then we have a whole bunch of other neurotransmitters that are not really discrete on-off signals, but they're more what we call neuromodulatory, which means they are changing the ability of those discrete signals to turn on and off. So they're regulating the brain activity, right? And so 2 of the ones that we're going to talk the most about today that I've already mentioned are norepinephrine and dopamine. Norepinephrine is released in the brain in response to arousal and is released in very stressful times to produce vigilance and heightened arousal in the brain. When you're sleeping, norepinephrine is very, very low. And so it's part of sort of our, like how awake we are, how stimulated we are neurotransmitter. So it's influencing those on-off signals. So when norepinephrine's around, the on signals are much more potent. When it's not around, the inhibitory signals are much more potent, which is one of the ways in which we're able to go to sleep. Dopamine is the one that we'll spend the most time talking about today. And it is used primarily in the basal ganglia and in the frontal lobe. And again, it's very much modulatory. It's sort of regulating the activity of those areas. And so you can imagine that if there's an abnormality, you don't have enough dopamine, for instance, then that regulation goes away and the system's not able to work efficiently and effectively. 2 other neurotransmitters that I'll just briefly mention. Acetylcholine is again sort of a mildly arousal neurotransmitter in the central nervous system. Also used to cause our muscles to contract in the peripheral nervous system. And then serotonin, which we know is involved with things like our emotions. I think we have a training coming up this year on major depressive disorder, and that's where we'll really dive into serotonin and its role in emotion and mood. But it also plays a role in sleep and appetite. And I mentioned serotonin because we don't fully understand the relationship between dopamine and serotonin. We're pretty good at, being able to go in and say, hey, this area of the brain that, for instance, controls appetite, we can see it has serotonin receptors, and we can see that there are neurons releasing serotonin here. So we know that that's how that it's doing this thing in this part of the brain. And if we block it or if we increase it, we can change the eating behavior of that animal. What's harder to understand is how does serotonin interact with other neurotransmitters? And we know that they sort of regulate each other, and activity in one system affects the activity in another system. And dopamine and serotonin have this type of relationship. And so when there are changes in one, it often causes changes in the other system. but we don't fully understand that relationship. It's not like if you have not enough dopamine, that causes you to have more serotonin. It's more complex than that. But we do know that these neurotransmitters influence each other at some level. All right, are we doing good with some of the nitty-gritty neuroscience stuff? All right, I see good head shakes and no head shakes and good head bobs. Lastly, in sort of talking about general how the brain functions, in addition to releasing those neurotransmitters as a communication device to the next neuron, or as a way to influence that neuron's activity in the case of the neuromodulators, we also, very importantly, need to remove that neurotransmitter once it does its thing. We don't want it to stick around. We want to remove it actually pretty quickly so that we get these small signals that are coming in. And there are three ways in which neurotransmitters get removed from the synapse. The first is that our body's very efficient at a thing that we call reuptake, but you can think of it as recycling, where we're grabbing the neurotransmitter with a protein, a transporter protein, and pulling the neurotransmitter back in to the presynaptic side so that it can get bundled back up and be used again. So we're not constantly having to make new neurotransmitter. We can reuse the neurotransmitter that our brain has already made. And so reuptake is an important way, particularly for dopamine and norepinephrine, that both use this mechanism to recycle neurotransmitter and get it out of the synapse. The second way is that we have enzymes that are floating around out here. And when these chemical enzymes come in contact with the neurotransmitter, they degrade it. They break it down so that it can no longer activate the receptor. So you can think about, you talk about detergents that have enzymes that are breaking down dirt and grime. Same thing here. We have these enzymes that are breaking down neurotransmitters and eliminating their action. Again, both dopamine and norepinephrine have enzymes that are breaking those down as well. And then finally, neurotransmitters can be absorbed by glial cells, which are support cells in the brain. And those glial cells can either transport the neurotransmitters back to the neurons for reuse, for recycling, or they can break them down and get rid of them in our blood supply. Dopamine and norepinephrine not so much broken down in this way. All right, so again, why do you care about this? Why is this important? Well, it turns out some of the ways in which we can develop drugs that can affect neurotransmitters involve blocking these systems, right? So if I develop a drug that can come down here and block the transporter protein and prevent it from recycling dopamine, that's going to increase the amount of dopamine that's present to activate that postsynaptic receptor there. All right. So going to delve a little bit into the development of the brain and plasticity. So I've used that word plasticity several times, and all it really means is that our brain's connections change over time. They change over time naturally with development. They change over time in response to chemical concentrations in our brain, whether that's natural chemicals like neurotrophins that I'll talk about, which support brain development and connectivity, or they can also change due to the continued presence of a drug. And that's what happens in addiction. I think we talked about addiction in our last training, where continued presence of a drug modifies connections so that the brain becomes dependent on the presence of that drug for those connections to work effectively. And then finally, experience. So as I mentioned, if you don't use connections, they're going to degrade and you're going to lose that connectivity. And if you use those connections, you can really improve their functionality. So in general, brain development, natural occurring brain development, begins about embryonic day 15, and it continues until your mid-20s. We now know that It doesn't seem like the brain is fully done its natural development until age 22 to 25-ish. That said, plasticity is happening throughout our entire lifespan, I hope, because every time that you learn something, if you're learning something right now, you're forming connections in your brain that are creating memories. And then if you rehearse that, then you can access those memories and they become permanent connections stored in your brain. So it's important to understand that while biology at some level does determine your brain's connectivity during development, once we get post-development for the rest of your lifespan, what's really determining your brain's connectivity is your experience and your behavior and how you choose to use those connections. And if you're putting any drugs in there that can alter that brain's connectivity. So there's really two things throughout the rest of your life that can alter your brain connectivity: how you use your brain and what you put in your brain in terms of chemicals. All right, so a little bit more on brain development. In the first 3 weeks, there's massive proliferation of neurons, and we can actually detect differences between the three major parts of the brain. The forebrain, the midbrain, and the hindbrain have already developed very early on. By 11 weeks, the forebrain is proportionally larger than those other parts of the brain, and is going to continue to develop and become by far the largest sort of part of the human brain. If you look down here at 6 months, we've now started to produce so many neurons in the brain. It is generating neurons in the innermost part of the brain, and those neurons migrate out to the exterior part of the brain. So the brain builds itself sort of from the inside out, and it keeps piling neurons on the outside of the brain, and that's growing the cortex bigger and bigger and bigger. And around 6 months, we've sort of reached the capacity of our skull to fill that whole area inside the skull with neurons. And so after 6 months, we're continuing to add these neurons. And in fact, we're adding these neurons at enormous rates, about 250,000 neurons a minute, and sometimes, so that we can get up to, by full development, that we have 86 billion neurons, in the brain. So after 6 months, the brain starts to create these grooves and folds because we're creating more surface area, because the skull is limiting the surface area. And so in order to generate more places to put neurons, we create these valleys called sulci that create these the wrinkled appearance there. And then at 40 weeks, we're full term. By 1 year, the brain very much looks like an adult brain and is showing electrical activity very similar to adult brains, although obviously there's still a whole lot of development that happens after 1 year of age. But sort of at 1 year of age, the brain's fairly stable, and it starts to do sort of more finite development as children learn cognitive tasks and learn lots of different things, social interactions, motor movements, all of those sorts of things. All right, so I mentioned neurotrophins and that these natural chemicals that are in the brain are what guides our natural brain development. And so, two things are happening. We're producing lots of neurons, but then those neurons have to go from this innermost part of the brain and find their destination. And they're guided by chemicals called neurotrophins that are being released from their target, where that neuron is supposed to be, and creating sort of a chemical trail that the neurons then follow. So neurotrophins do two things. They help the neurons migrate and get to where they're supposed to be, and each undeveloped neuron is seeking a particular type of neurotrophic chemical to get it to the right place. And then once they get there, neurotrophins help increase connectivity between neurons. And so they help cement those they actually help the neurons differentiate into the right kind of neuron, and they help them with their connectivity, right? And so down here in this clip, you're going to see a bunch of, this is actually in a culture dish, but those are undifferentiated neurons that are migrating. And we have two sources of neurotrophins. We have neurotrophin 1 up here, and you can see a bunch of neurons that are sort of reaching out, seeking towards that area. And then we have neurotrophin 2 over here, and we see another group of neurons that are attracted to that neurotrophin. And right here I've highlighted, you can see one neuron starts going towards neurotrophin 1, and then it realizes, no, that's not the right chemical signal. And so it turns and heads to neurotrophin 2. I say this because chemical signals in the brain are very important in terms of getting neurons to the right place and then causing them to become the right neurons and form the right connectivity. Here we see two neurons that have now differentiated, and these two neurons are using neurotrophins to find each other, right? And they're going to form a connection here and form a synapse. And so again, all of this is happening through those chemical signals. And so now we see that those neurons have actually reached out and formed this synapse here. There's actually a smaller one over here as well. So initially, this brain development happens based on this natural blueprint for neurotrophic support. But then it's experience that is strengthening these connections and causing more connections to occur, or the lack of experience that causes those connections to deteriorate. So that neurotrophic support really only happens in that first developmental phase. Again, by about 3 months of age, we have generated all the neurons that the brain is going to need. And what really happens after that is the connectivity between those neurons is changing, and it's changing through experience. All right, so we can talk about sort of 4 different developmental stages. We have a very, so we have our sort of in utero development here, where we get putting out lots of neurons with lots of synapses. And then we have a phase between when the brain becomes stable at about 3 months of age and it's not really making new neurons until sort of early childhood. That is our initial sensory motor learning. And here the brain tends to produce more connections that it needs. And then through learning and development, we're pruning out the connections that aren't correctly involved with that ability to move or ability to perceive our sensory input. And the ones that are being used correctly to do those tasks get strengthened and remain. So there's sort of this pruning effect where we're getting rid of the connectivity that isn't appropriate and strengthening the connectivity that is. And then from about 3 years of age to adolescence, we have a lot of learning that's happening, right? So children are learning complex cognitive tasks. They're learning about theories of self and self-awareness. They're learning social interactions. They're learning how to read. They're learning a lot of complex things, right? They're learning language at that time. And so there's, again, a big pruning that happens there where we're eliminating unwanted connections and strengthening the ones that are doing the stuff that we want to do. And then we have actually a period where we increase the connectivity of the brain. And this happens in that final developmental stage in adolescence. And so here we have sort of, by early adolescence, learn most of the things that we need to survive in life. And so now we're sort of fine-tuning things, right? And so we're kind of crystallizing or cementing those connections that are there. And a lot of that happens through strengthening of connections. And so we see actually some increasing in synaptic connectivity during that time. And then, as I mentioned, after about age 25, we have what we call lifespan development. And unfortunately, that's about your peak there in terms of neurons. It starts to go downhill. And so we do, over time, start to lose neurons in the brain. And when we lose neurons, we can't regain them. So while connectivity can change in the brain, as we age, we do unfortunately lose neurons. And it's why, if you're fortunate to live to be very old, in your 80s, you're going to have some memory issues and some movement issues. You're just not going to move as fast or have as quick reaction time because we've lost some of that ability. All right, so going back to the changes that happen in that adolescent maturation. And this is the time at which, sort of age 5 through age 20, that we see the onset of ADHD often. It's in this sort of final crystallization phase that those deficits, we call them deficits, superpowers become observable. And so here in these, this is a video that I'm going to play. We see several different, we see a image of the right side of the brain, image of the left side of the brain, the top of the brain, and the base of the brain. And the areas that are in red are the least developed and least connected parts of the brain. And the areas that are purple represent the most crystallized and sort of developed parts. And so this is as we age from 5 to 20. We see the main thing that's happening late in adolescence is all of that purple coming in, right? And so here at about age 11 or 12, not a whole lot of purple, not a whole lot of connectivity. And then in that last sort of burst of the teenage years, we get that connectivity that happens. And these are just some time snapshots that you can just really see that connectivity. And the last part of the brain, right? Most of the brain here has developed its connectivity except for the frontal lobes. And the frontal lobes are sort of the last part of the brain to come online to get sort of crystallized. Why do we care about this? What does this mean? That children have less connections than adults do. Well, this is a study that looked at brain imaging, looked at brain activity. They gave them a working memory test, and they did this in children here, and they did it in adults. And if you look, the same sorts of areas of the brain are being activated in both children and adults. But what do you notice about the brain activity in the adults compared to the children when they're doing this test? Does anyone see differences? All right. I'm not going to make y'all be interactive, but if you look, you can kind of see that there's less activity in the adults than the children, right? They're having to use more brain activity. In fact, if we quantify that in a graph, you can see, for instance, in these particular areas, that in order to do this working memory tasks, the adults need less brain activity than the children do. So when we say this task is more difficult for children, it means that they're having to devote more brain processing power in order to do that because their brains aren't as well connected, right? They're not able to, they're having to work harder because they're not fully developed yet. As I mentioned, experience also can affect development. So here we see, this is now EEG recordings from some different locations in the brain. And these are teenagers that have been grouped into low video game exposure and high video game exposure. The low video game exposure, I think, was less than 5 hours of video games a week, and the high was greater than 35. So a lot of time in front of a screen playing games. And what we see, and I don't have to get into the specifics of what these differences in the brain activity mean, but we see in response to the same stimuli that their brains respond electrically in different ways. And this, we presume, is based on the fact of the video game exposure, since we've grouped them into those groups and tried to control for other confounding factors. And in fact, when we give them two different types of tasks, a proactive task where they have to make decisions based on internal thoughts, and we give them reactive tasks where they have to make decisions based on external stimuli. The low gamers tend to be better at the proactive tasks than the high gamers are. And the high gamers tend to be better than the low gamers on reacting to external stimuli. So it's almost like the massive exposure to the external stimuli of video games have trained their brain to be more responsive to external stimuli. And probably because of that fact, and we have limited resources, it's sort of taken away some of the areas of the brain that are responsive to internal stimuli, the proactive decision making. All right, so the whole point in talking about brain development is to talk about, do HDAT brains develop differently? And the answer is yes, they do. And the question then, the million-dollar question is, well, why do they develop differently? And we certainly know that there's a host of genes that are involved with shaping brain development. We're less sure that experience, that some environmental effect causes ADHD, or some chemical exposure causes it. There's less sort of evidence to sort of understand that. If we look at just quantitatively from a neuroanatomical standpoint, ADHD brains versus control brains that don't show ADHD symptoms, ADHD brains overall tend to be slightly smaller. Their volumes are smaller, and in particular brain structures, there appears to be a lag in the developmental timeline of the brain that somewhat catches up by adulthood. We know that there are specific brain structures that are affected, and those are areas like the frontal lobe, the basal ganglia, and the cerebellum. And we'll talk about what that means today. All right, so here we have two structures, the putamen and the caudate, that are part of the basal ganglia. The basal ganglia, as I mentioned, is involved in sort of regulating brain activity. And if we look here, this is bins of ages. And if we sort of separate that into a childhood phase and an adolescent phase, what we see is that in childhood, the ADHD is slightly smaller than the controls for both of these areas. Yet for these areas, by adolescence, they generally catch up. And throughout adulthood, they're pretty much tracking in terms of the same volume there. Again, this is just descriptive. This is lots of brain imaging studies that have just measured the sizes of these structures. We know that's sort of easy to do. What's a lot more complicated is to say, well, what does that mean? What does that mean in terms of cognition? What does it mean in terms of how connectivity is developing in those areas? And that's sort of where we know less about that. Over here, we have two structures that are involved with reward. And what we see here, again, if I put up those boxes, is that we have differences here in childhood that persist into adolescence, right? And somewhat sort of catch up again by the time that we get to adulthood, right? And so we'll talk about two specific theories of ADHD that are involved with the reward system that have to do with how we learn things. So we learn things because we get reinforced for doing those things, and things that we get reinforced for doing, positively reinforced, we're going to form stronger connections and learn those things. And so being able to associate a reinforcement with some action or thought or behavior is very important. And what we see is some deficits in ADHD clients that have a harder time linking that reinforcement with reward, that make it more difficult for them to learn things, to learn these, to form this connectivity. All right, I see a couple of questions in the chat, and so there was a question about what do neurons look like for drug addicted mom and baby? Yes, so certainly babies that are exposed to chemicals in uterine, that can affect brain development. The good news is that, as you saw, a lot of that brain development occurs postnatal. And so getting those drugs, those babies weaned off the drugs after they're born, helps the brain develop normally. But often we see delays in learning and learning disabilities in babies that were exposed early on to particular drugs, and that can affect it. Message about adults that are slow processors. Sure, I mean, everybody has individually developed brains, and some people's brains just may not form as strong or as many connections as other people's brains, and that might underlie that. Again, this is some complex stuff that's fairly difficult to do research on, and so a lot of this is sort of theoretical in nature, right? And data on this for medicated versus unmedicated kids. I'll talk a little bit about that later when we get to the medication parts. But a spoiler alert, there's not a whole lot of research on this, unfortunately. And you would think that there would be a lot more research in this field, but I'll talk about some of the reasons why there isn't as much research on this. All right, we are coming up on an hour. I would say let's take a little bit more than 5-minute break. Let's come back at 5 after the hour and start back up, and let everybody turn their screens off for a few minutes, get a drink, use the restroom, whatever you need to do. And I'll see you back here 5 after the hour.
Thank you. See you guys in a few.
All right, so we're doing okay, not glazed over yet. We got so we're going to jump into some of the ADHD specific stuff right now, and I'm not going to spend a whole lot of time. I'm going to focus most of my time on neuroscience and that aspect of ADHD, as opposed to stuff that you all I'm sure know a lot more about in terms of how you do your therapy and how you diagnose and that sort of stuff. But just to go over the basics of ADHD, there are two primary conditions: inattention and the hyperactivity and impulsivity. And you have to have 6 or more of the symptoms that I'll talk about on the next slide, and those be present for 6 months. It also, again, is a developmental disorder, so we want to see evidence that these symptoms were present prior to age 12 in that childhood developmental area timeline. And that these are present not just localized to a particular environment. So it's not just that the child is having a difficult time at school, but that these are also being shown in the home as well. And they have to be debilitating, right? So, everybody has, tunes out things and can daydream and that sort of thing. But If it's becoming interfering with your ability to function, then that's when it becomes a diagnostic criteria. So the inattention criterion, there are, you need to have 6 of these 9. And it's a lot of the things that we heard Penn talking about in his songs earlier, right? So making careless mistakes, difficulty sustaining attention, doesn't seem to listen, not following through, again, on schoolwork, losing things, not keeping up with things, being easily distracted by external stimuli, forgetful, difficulty organizing, and reluctant to engage in tasks that are what we call goal-oriented tasks, right? That are going to require multiple steps or require you to sustain some sort of process to get to the completion of that. The hyperactivity criterion, again, 6 or more for at least 6 months, fidgets, unable to stay in their seat, on the go, talking excessively, interrupting, difficulty waiting for their turn, those sorts of situations. So what do we know about clients with ADHD? Well, the prevalence is around 5%, both in childhood, in age under 18, 3.5%, in 18 to 44-year-olds, slightly less. So not everyone persists into adulthood, and some adults get diagnosed that weren't previously diagnosed in childhood. You know, some parents may be reluctant because of stigma or lack of education or lack of resources to get their children diagnosed. And so it's not until they're out on their own in adulthood that they're noticing these problems that they go and see someone so that they can be diagnosed. So there are some changes there. This is not equally distributed across the country. So there are some states with higher amounts of ADHD. And I know y'all are in Alabama. That's one of the 4 highest states with a prevalence about 3 times the national average, around 15% diagnosis. It's much more frequent in males than females. So a 3-to-1 ratio, 3 times more males than females. We're not exactly sure what this means. There might be some evidence that estrogen could be protective. There's a lot of sort of neuroprotective effects of estrogen. And particularly with the dopamine system, there may be some interactions there that make males a little bit more susceptible to this. Females tend to primarily present with the inattentive features, whereas males tend to also have that hyperactivity. Not to say females can't have hyperactivity, but it's it seems to be a sex difference there in what they present primarily with. As I mentioned, 60% of ADHD symptoms persist into adulthood. So this does it's a developmental disorder that becomes a lifespan issue that people have to continually deal with. And the inattentive symptoms are the ones that tend to persist. So often ADHD children sort of grow out of the hyperactivity, or they learn how to better cognitively control that. And it's maybe more difficult to harness that inattentive symptoms with age. What are some of the things that we know correlates with ADHD? We know that children with ADHD are more likely to develop conduct disorder and antisocial personality disorder in adulthood. And conduct disorder in adolescence is related to antisocial personality disorder in adulthood. So but there's some relationship there. And it the hypofrontality, the fact that the frontal lobes are not functioning at their highest level, can lead to behaviors that result in things like conduct disorder, right? Not understanding the consequences of your actions, or not caring about the consequences of your actions, or not understanding how to inhibit behavior that's inappropriate. Also, this doesn't just affect learning in the classroom or work or classroom activities, but it also can affect other complex behaviors like driving. And so if we look at newly licensed drivers, ADHD drivers have a much higher rate of having a crash within the first 48 months of having their license, both males and females, although males have an even more likelihood of having a crash than non-HDA newly licensed drivers. So this can translate into other areas. There's also comorbid anxiety and depression, often associated with ADHD. And people often identify the ADHD as the source of that anxiety or depression. So, having stress about a test, having test anxiety because they're not able to concentrate or they don't think they're going to be able to do well on that test. That can translate certainly into adulthood, where often we see particularly in women that in adulthood that have ADHD, a lot of the times they have guilt and depression that they're not doing their best at being a mom or taking care of the house because they're having trouble with goal-directed behavior to do those things. And so often this is where counseling can really come in, because helping individuals identify the source of that and doing neuroeducation and saying, look, you don't really have depression, major depressive disorder. You're feeling depressed because you're not able to do these tasks. Let's work on treating the ADHD so that we can eliminate this guilt or these anxiety feelings that you have. All right, so getting into sort of the underpinnings of ADHD. And so when I present these things, this sort of technical data, I want to try and cite my references on the slide. So usually at the bottom of the slides, you'll see links to the actual research publications that are supporting this information. And so, as I mentioned, we know that there are multiple genes involved. There is a strong heritability between parents and children with ADHD. If one sibling has ADHD, there's a 9-fold increased risk that another sibling in that family will also have ADHD. And if we look at identical twins, 74% of the time, and one identical twin has ADHD, the other identical twin also has ADHD. So definitely a strong sort of genetic component to ADHD. As I mentioned before, there's a reduction in the total brain size, and specific structures are involved. So there is less connectivity in the prefrontal cortex white matter. So the brain, the neurons in the brain, we talked about the neurons in the brain being the dendrites and the soma, or the cell body, which is involved with processing the signal. And the dendrites and soma are found in areas that look gray in the brain, and we call that gray matter. The output is the axon, and those axons are myelinated. They're insulated with actually a fat cell that we call myelin in the brain, and that fat cell is white in color. And so anytime we see white matter in the brain, we're really talking about those axon connections to other neurons and other parts of the brain. And so less white matter means less connectivity within the frontal cortex and between the frontal cortex and some other areas of the brain, like the basal ganglia. And so we already mentioned in that previous slide that there's a developmental delay in 2 areas of the basal ganglia that receive input in the caudate and the putamen. And in general, there's sort of weakened connectivity within the basal ganglion. And I'll talk more about what the basal ganglion does in just a minute. As we saw in that slide, there's a developmental delay in both childhood and adolescence in areas involved with the reward network. And those components end up looking smaller in ADHD brains. And then finally, in terms of neuroanatomy, we see a reduced size of the cerebellum. And the cerebellum is, again, linked into motor. And so some of that hyperactivity may be a result of the cerebellum not being able to control that motor hyperactivity system as well. And it also has to do with timing and associating rewards or reinforcements with stimuli, that sort of thing. Sort of that basic operant learning that happens in the brain. Lastly, it looks like there are reduced levels of that neuromodulatory neurotransmitter dopamine, particularly in the frontal lobes and the reward systems. Right? And I'll talk a little bit more about that. But I want you to sort of just keep these things in mind. In fact, this would probably, should have said this before I went through it all, but maybe let's take a second and just jot down like decreased prefrontal connectivity, smaller basal ganglia, smaller reward, smaller cerebellum, right? So when we talk about what those particular parts of the brain do, you'll know that there's this neuroanatomical correlate of ADHD brains that we don't see in these other brains, in control brains. All right, so what is executive functioning? I sort of touched on that very briefly when I went over the role of the frontal lobe. The frontal lobe, prefrontal cortex, can be broken into sort of two bigger categories. The dorsolateral prefrontal cortex, which is the top part of the brain shown in pink, frontal lobe, and the orbital frontal cortex, which is sort of the ventral part of the brain. And these two parts of the prefrontal brain do some fundamentally different things. So in the upper part, the dorsolateral prefrontal cortex, that's where we have our attentional control system. All of these are part of what we call executive functioning, which is sort of our highest cognitive activity. And attentional control is our ability to concentrate and focus and maintain focus. And along with that is where our working memory is stored. So our working memory, or sometimes referred to short-term memory, that's sort of an older term, we now sort of refer to that as working memory, is the place in our brain where we are actively using information. And so while working memory is not, we don't see deficits particularly in working memory in ADHD brains, but what we see is deficits in the attentional control of working memory. So if you're not actively using that information in working memory, then it goes away from our cognition. And that's often what we see is this shifting, this inability to stay focused and continue processing and working through a problem or a task using our working memory. The lower part of the brain, the orbital frontal cortex, is involved with what I would say is like more sort of basic processing. So a thing called cognitive inhibition, which is our ability to tune out interfering stimuli. So we're constantly being bombarded with other stimuli, and in order to stay on task, we have to recognize what's appropriate and not appropriate to pay attention to. So that's different than ability to concentrate. Here we're saying what's going to interfere with that ability to concentrate. And then inhibitory control. And this is the ability to inhibit impulses, to determine inappropriate behaviors and suppress them before they are expressed, and delayed reinforcement, right? So saying the ability to say, okay, I'm not going to eat the cookie batter now because I want to bake the cookies and have them as a done product. And so I'm going to delay that reinforcement until they're done cooking in order to have that reward, right? So sometimes we have to avoid short-term rewards in order to have a goal-directed behavior become accomplished with a reward at the end of that goal-directed behavior. And then finally, the entire sort of frontal lobe is involved with this thing called cognitive flexibility, which is the ability to switch between different modes of thinking and to process multiple concepts simultaneously. That involves things like planning and motivation, reasoning, problem solving, measuring consequence, all of those sorts of things. All right, someone asked if the executive functioning is similar to serving as the secretary of the brain. It's really sort of the, yeah, it's like the control center, if that's what you mean. It's sort of guiding general brain activity, particularly cognitive thought. And so how do I use those perceptions, right? If I see a squirrel, am I going to be distracted by that, or am I going to use my cognitive inhibition to tune that out because that's not important to what I'm doing right now? All right, so in order for the frontal lobes to work efficiently, they're going to be doing a lot of discrete signaling with glutamate and GABA. That's how they're processing information and forming memories and making decisions. But that has sort of this overarching neuromodulation that's supporting that. And this is done through both dopamine and norepinephrine in the frontal lobes. And we need an optimal level of these neurotransmitters. And if we're suboptimal, then that's not good. And if we have too much stimulation, that's also not good. So there's sort of a sweet spot, and we call that an inverted U-shaped curve. So that we see if we have too little stimulation of norepinephrine and dopamine, we get poor impulse control, poor inhibitory control, and we get unguided attention, right? If we have just enough, then we have guided attention and goal-directed behaviors in all of those areas, the inhibition of interfering stimuli, the suppression of impulsivity, the delayed reinforcement, all of that works together so that the frontal lobes can do their cognitive tasks, right? If we have too much dopamine or particularly norepinephrine, then we get cognitive inflexibility where we're not able to do to shift between those modes. We get hyper-focused on a particular thing, and that then interferes with our ability to, we have that misguided attention on the inappropriate things. All right, so getting into the basal ganglia and how it interacts with that executive control network. So the basal ganglion helps regulate the activity that's going on in the frontal cortex, and it actually helps regulate a lot of systems. It's not just the frontal cortex, although that's certainly one. So there's a motor, so there's these feedback loops that come out of different areas of the brain, run through the basal ganglia, and then the basal ganglia gives the appropriate output back to the area where the input came from in order to make everything work right? And the motor system runs through the basal ganglia to help regulate our motor activity. And in Parkinson's disease, we lose this area here called the substantia nigra. That's what's deteriorating in Parkinson's disease. And we lose the dopamine input into the basal ganglia, and that results in our inability to regulate motor outputs. So you get the rigidity of Parkinson's disease. You don't have fluid movements, so you get shuffling walk, the inability to suppress tremors, and so people will have tremors. And eventually, that transitions with the deterioration of that disease from just being a motor disorder, which are the first symptoms, into some of these other areas like emotion. So emotional regulation goes through the limbics, goes through the basal ganglia. And so again, Parkinson's is an easy thing to identify because we know very clearly what is degenerating and how it's affecting the basal ganglia pathway, but it gives us insight into what the basal ganglia does when it's functioning normally. So normally the basal ganglia helps regulate our emotion. In fact, in obsessive compulsive disorder, that is also a disorder of the basal ganglia where we're not able to regulate those emotions or those thoughts. And then the prefrontal cortex, as I mentioned, also is running through here. And so it provides this feedback loop to help us have correct executive control, correct activity in that network of executive control. All right, so this is what the basal ganglia kind of looks like if we diagram it and diagram the connections. And what we see is we have the frontal lobes here, cerebral cortex, that are coming into the caudate and putamen, which we already said in childhood show that developmental delay in ADHD. So that's the input coming in here. And then overall in adulthood, this entire area is going to have less of these appropriate feedback connections, and it's going to be smaller in size. Dopamine, again, is neuromodulatory. It comes in and it provides this regulatory role that helps this feedback loop do what it's supposed to do. And as long as we have sufficient levels of dopamine in here, then the basal ganglia is working well and it's providing the right feedback to the frontal lobes. But when we don't have appropriate control, it's all of these GABA and glutamate pathways that are in here are sort of unchecked, and it's not able to provide the correct outputs back to the frontal cortex. And so this is part of how we think the basal ganglia and dopamine are involved in ADHD. I also mentioned the reward system, and so in addition to the amygdala here and the nucleus of accumbens here in the forebrain, there's this cortical area that's on the midline right above the corpus callosum, and it's called the cingulate cortex. And this is part of that emotional regulation. And often, some of the symptoms of ADHD is impulsivity and emotion and lack of emotional control, that interruptive speech, right? So more of the hyperactive impulsive symptoms might be involved with this circuitry. And the fact that that those symptoms tend to not persist as much into adulthood could be that developmental delay sort of catching up to in adolescence and into adulthood. The nucleus of accumbens, as I mentioned, it uses dopamine in that system. So we're seeing dopamine as sort of a reoccurring theme here in terms of a neurotransmitter that's involved in multiple of these systems. And dopamine is our natural reward system. So when we do something that makes us feel good, that good feeling comes from dopamine being released in the nucleus of accumbens. And then the amygdala is, again, assigning emotional value to stimuli and providing motivation in response to stimuli. And so there are signaling pathways, this frontostriatal, there's signaling pathways between the nucleus of accumbens rewarding correct decision-making in the frontal lobes and the amygdala giving appropriate value to internal and external stimuli. So when we talked about the high video gamers and low video gamers valuing internal stimuli versus external stimuli, that's part of what the amygdala is doing, and changes that have happened in the amygdala because it's been shaped by all of that experience of responding to external stimuli with the high video game play. All right, and then sort of the final network that's involved. So far we've talked about specific role of the frontal lobe and executive function, and that executive function is regulated by the basal ganglia, right? And that we have this other network that's involved with emotional regulation that also then affects attention and affects our ability to have correct values for internal and external stimuli. The final network that's involved in this system is the reward network. And here, this is where we get into sort of the inhibitory control or the impulse control. So the orbital frontal cortex, it's sort of shown a little bit higher in this brain diagram, but that was the blue part of the frontal lobe that's in the lower area, is responsible for this, and it gets input from both the ventral striatum, the basal ganglion, and the amygdala. And dopamine is the main sort of neurotransmitter that is setting the stage for this connectivity and these pathways to function properly. This is where we have what we call the delayed reward system, where we're inhibiting taking a smaller reward in order to accomplish a bigger goal that presumably would give a bigger reward or a more important reward. So it's not necessarily that the reward's bigger, but that we value, completion of the entire exam and not just the first half of the exam as the overall goal, right? And this is what helps us complete complex goal-directed behaviors. And that's going to lead us into the first of our sort of biological theories, theoretical causes of ADHD, which is dynamic developmental delay. All right, so we're going to jump into there. We're going to have, so I've talked, that last little section was on the neurobiology that could be underlying it. What networks are involved with ADHD, right? So executive function, control of executive function by the basal ganglion, emotional regulation, and reward systems. And so there are several different models or sort of theories about why we have ADHD. And the first one is this thing called the dynamic developmental delay theory, or delay aversion, it's sometimes called. And it can be a little complicated. I'm going to try and break it down and make it as simple as possible, right? So when we bring ADHD clients in for research testing and we measure their ability to learn and respond to different tasks, we see that they demonstrate this thing that's called a steeper delay of reinforcement curve. So here, the normal delay of reinforcement curve is shown in the dashed line over here. And this steeper curve here is what we see with ADHD. So what does this mean? Well, overall, it means that a reinforcer, which is this RFT here at the 0 point, or this point over here where we see the y-axis go up, right? A reinforcer loses its value. The effect of the reinforcer becomes less and less and less in a quicker time for ADHD patients than in normal controls. Why does this happen? Well, we think that dopamine is important in being able to sustain that delayed reinforcement. And so we're going to talk about two different ways dopamine is released in the brain. Tonic release of dopamine is sort of a rhythmic, constant bathing of the frontal lobes in dopamine to regulate, to do that neuromodulation. So when we talk about there needs to be an optimal level of dopamine, that's this tonic release. Then we'll talk about phasic or burst type release in response to specific stimuli. So when we get that reward, that's a burst of dopamine that causes that pleasure feeling. So we think that this part, this delayed reinforcement, is due to not having enough sort of constant levels of dopamine in the brain. The result of that is that often in research settings, ADHD clients want to take smaller and immediate rewards over a larger reward that has to be delayed. And so if we have a response here, like response C, that's close to the reinforcer, so we're going to have a response, and then within a few seconds, the person is reinforced, then we see the effect of that reinforcer is very high, right? Because the response happened close in time to the reinforcer. And if we move that reinforcer back in time, so say now it's several minutes between this response and the reinforcer, in the normal brain, that's still producing a decent effect of that response, of that reinforcer on that response. But this steeper curve here results in a much smaller effect of that reinforcer for this response. And in fact, we can have responses that are still somewhat reinforcing in a normal brain, but lack any reinforcement. So here we're beyond this effect of this curve. And so an ADHD brain doesn't make the connection, doesn't make the normal connection between, hey, that thing I did 10 minutes ago led to this eventual reinforcer that happens. So this makes it, again, makes complex goal-directed behaviors difficult, or what we say, staying on task. They can't put the early steps, those responses, in touch with the later reward, right? They don't get, those connections don't get strengthened in the brain because there's not that association that's happening, because there's not enough dopamine to sort of regulate that. The second theory is what we call dopamine transfer deficit theory, and it's also involved with this delay aversion, right? And this has to do with that phasic, or that burst of dopamine that's released. And essentially, when we have that reinforcer happens, we get that burst of dopamine. And when a response happens frequently enough with the reinforcer, our brain learns that that response actually becomes reinforced by our brain. And we call that anticipatory dopamine cell firing. So here we have an unexpected reward. There wasn't something paired with that reward, and we get this burst of dopamine in the brain. Now we pair a cue with getting that reward. And if we do, and if we do this enough times, the cue starts to take on the actual reward. And so once that's learned, our brain actually releases this anticipatory dopamine reward response when you do the thing that's going to lead to the positive outcome, right? So in a complex task like learning how to study for a math test, right? The more times that you invest study time correctly and it results in the reward, like the first time you do that, you get the reward of, hey, I got an A on that math test. That made me feel really good. And the more times you do that, eventually the studying in a normal brain takes on a reinforcer. You're like, yeah, I don't really want to study, but I'm going to study because I know I'm going to get this reward. And so it takes on, that cue takes on the actual reward because you know that it's going to happen. And the brain will motivate you to study by giving you this anticipatory dopamine cell firing. Right? So cues before the reward actually become so associated that they can cause dopamine to be released in the brain. And this is something that we can study, like these basic sort of components and how this works. We can study in animal models. And so we can actually measure dopamine release in rats that learn to press a lever and get a reward. And after this learning happens, when they press the lever, the brain is being stimulated, not when they get the food reward, right? So we can actually see these things in animal research, right? In ADHD brains, what is theorized here is, and what we tend to see with their behavior, is that it's hard to transfer that dopamine from the reward to the response, particularly, again, going along with that steep curve. If the response is studying the night before, long delay until you get your grade 2 days later, it's very hard to transfer that dopamine release in these brains. And so that then takes away the motivation, right? And makes it difficult to maintain on-task behaviors. This can also, we can think about this with hyperactivity, right? So children are asked, to remain in their seats in the classroom, and the reward for remaining in your seat is something that happens, a lot later when you get praised or you don't get punished, for instance. And in the inability to associate the fact that I'm staying in my seat means I'm not going to get punished or I'm going to get a reward, may be a difficult thing for someone who's not transferring that dopamine to that cue to maintain. Right? All right. The next theory is a brain circuit sort of control theory of why ADHD happens. And this involves those sort of three general areas of the brain. It says that there are dysfunctionality of the prefrontal lobe to exert its control, and this is partially due to dysfunction in the basal ganglia to be able to help the prefrontal cortex tune out interfering stimuli and inhibit impulsivity. So the frontal lobe is in general not doing a good job of executive function, and it's not being helped by the basal ganglia in order to regulate its activity. And then the third part of this is that we know that the cerebellum, again, smaller in size in ADHD brains, the cerebellum has connections both with the basal ganglia and the frontal lobe. And so we think that dysfunction in this pathway leads to problems predicting when events are going to occur and with the timing of those reinforcers. So this model sort of helps explain the first two. So why is there this steep reward reinforcement curve, and why can ADHD brains have a hard time associating cues that occur earlier with the reward? Is that the cerebellum's not able to give the brain the timing information that it needs in order to do those things, right? And so we can see how ADHD symptoms, right, the inattentive executive function dysfunction, the impulsivity and hyperactivity of the motor system can be a result of cognitive control, reward processing, and timing that are all associated with these three different circuits or pathways that are happening in the brain. All right, and so finally, there's this, the sort of overarching model of not enough neurochemicals in the brain, the too low neurochemical level sort of model. This is almost exclusively based on evidence that if we give drugs that increase these neurotransmitters, that it improves ADHD functions. Therefore, it was a lack of these neurotransmitters that were causing the symptoms. That's really the main piece of evidence that we have for this. Now, I will say that the effectiveness of those drugs and their timeline of effectiveness supports a chemical imbalance model, a model that says there's not enough of these chemicals. So for instance, when a child with ADHD takes their drug, within an hour, their behavior has changed, the ADHD symptoms have decreased, and when that drug's effectiveness wears off, the symptoms come back, right? So it really is a good piece of evidence to say increasing this neurotransmitter is what's improving it, and not having enough of this neurotransmitter is what's causing it. That's not the case in other types of medications like antidepressants. Antidepressants often take days and weeks to have their full effectiveness, even though they're changing serotonin levels immediately, just like these drugs are when you take the pills. So this is a case where it does look like not having enough dopamine or norepinephrine is sort of the case. And if we look at the genes that we've been able to identify They also support this model because most of the genes are involved with dopamine and norepinephrine or noradrenaline as the two sort of neurotransmitters that are involved in this that we've been mentioning. All right, we're doing good. We're gonna get through a little bit more and I'll give you another break. All right. So we'll talk about these neurotransmitters, and then in the final sort of section, we'll get into the pharmacology of what's going on there and are there long-term effects of these drugs on the brain. Dopamine as a neurotransmitter, we know, like these are the easy things that we've known for quite a while. The neuroanatomical, the connectivity, the release of these chemicals. There's two major sources in the substantia nigra, which is what deteriorates in Parkinson's disease, and the ventral tegmental area, which substantia nigra is sort of the main contributor of dopamine to the basal ganglia area that we talked about that helps regulate that control. And the ventral tegmental area is the main supplier of the amygdala and the frontal lobe areas. And interestingly, you see this line right here, right? It really is sort of the prefrontal cortex that's getting this tonic bathing of dopamine. And that's different than norepinephrine, which is going to coat the entire brain, or something like acetylcholine that's also going to coat the entire brain. This really is sort of a localized signaling molecule that's needed by the prefrontal cortex to do its job. So as I mentioned, three different destinations primarily, with this specialized area of the prefrontal cortex or the orbital frontal cortex, the nucleus accumbens being that specialized reward center, right? All right. Two different sort of firing modes that I mentioned. That tonic is sort of a clock-like rhythmic bathing of the cortex with dopamine that's going to help regulate neural signaling in the executive function areas. Right? The phasic firing release is burst-like firing in response to a particular stimulus. So that reinforcement creates that burst-like, or the cue that has become associated with further reward down the line gets that burst-like firing that sort of reinforces this is a good thing, motivates you to continue doing that behavior. A couple of things about this system in the basal. So we have, as I mentioned at the very start, 5 different classes of dopamine receptors. So dopamine can activate all 5 of those. Right? Each of those 5 do different things. So for instance, in the basal ganglia, the D1 receptor is excitatory and the D2 receptor is inhibitory. So the same neurotransmitter in the caudate is inhibiting, but in the putamen area is exciting, because there are 2 different receptors in each of those areas, even though it's the same neurotransmitter that's coming in. In the prefrontal lobe, that tonic activation is through the D1 receptor. And what we think that that is doing is helping decrease noise by weakening connections that are not in use by the executive function systems. So when dopamine's doing its sort of normal role in the frontal lobe, it's helping the executive function circuits that are in use, right, that are being appropriately used, become stronger, and it's blocking other connections that would interfere with that process. If we have excessive, so again, we talked about that optimal curve, right? If you have too much dopamine, it actually impairs the circuits that should be able to be used, right? And so it's not just impairing the ones that are not in use, but it's impairing the ones that are in use, and sort of globally weakens the executive function center. And that's where we get that lack of cognitive flexibility, and people get stuck on one particular mode of thinking or one particular misguided attention. A couple of things I want to point out is that our brain is really good at monitoring how much it's stimulating other parts of the brain. And one of the ways it does that, as you see here, this is the dopamine that's being released to this postsynaptic neuron. There's a D2 receptor that we would call an autoreceptor that's basically measuring here, how much am I stimulating this neuron over here? And so it's a feedback loop to say, hey, I know that this much dopamine that I'm releasing is providing this much input to this other neuron. And it's being very efficient at recycling and pulling that dopamine through the transporter protein back into the synapse. The reason I mention this is that when we start talking about drugs, if we come in and we block this dopamine transporter, that's going to mean that more dopamine stays in the synapse. But it also means that this neuron's going to say, hey, I'm overstimulating this receptor, and so I need to cut back on the dopamine that I'm releasing. So there are compensatory mechanisms that neurons use to sort of stay at status quo, even if that status quo is not enough dopamine. The neuron doesn't know that it's not releasing enough dopamine, right? It just thinks that this amount of low dopamine is what I should be doing. And if a drug comes in and starts manipulating that, I'm going to try and compensate for that. And we'll talk about that. All right. I see a couple of questions in the chat. I will get to them when we get to a good stopping place here. Norepinephrine. So this says noradrenaline. Noradrenaline is kind of the English nomiker for norepinephrine. We tend to use norepinephrine in America more. It's just, they're the same thing. It's just two different words that mean the same neurotransmitter. So again, there are multiple different norepinephrine receptors, and it appears this particular type called alpha 2A is used to improve functioning in the prefrontal cortex and strengthen neural connectivity. And when we have and so there's an optimal level again of norepinephrine that stimulates that. And when we have too much norepinephrine, it begins to activate this alpha 1A, which actually has the opposite effect and can impair cortex function. All right, so overall, we can think about this as like these two neuromodulatory neurotransmitters are fine-tuning the activity in the executive control system such that norepinephrine is strengthening it and dopamine is reducing the noise that might interfere with it, right? And that's how we think these two, in a normal brain, these two things work. If we have too low levels of these, of either one of these, noise becomes more apparent, right? Or we're not able to increase the signal sufficiently. All right, couple more things and then I'll give us a break. I've mentioned genetic contribution several times, and genetic understanding genes and their role in development and in sort of control of the brain can be pretty complicated. So I just want to break this down and make it a little bit more hopefully accessible. So the names of these genes aren't really important, but I think it's important because to understand this, because it shows the connection between how heritability or genetics can influence these systems. So there's a gene, DBH, it's involved in making dopamine molecules. So if you have a problem with that gene or an allele that gene doesn't work as efficiently, you're not going to make as much dopamine in the brain. This DAT1 has two different functions. We know that it influences the size of the caudate, which again was one of the structures that we wrote down and said has smaller volume in ADHD brains. So this gene not working efficiently can cause that reduction in the basal ganglia. It's also the gene that makes the transporter protein that's going to recycle that dopamine, right? And these are all genes that we see differences in their expression in ADHD brains compared to control brains. That's how they've been identified. There's a gene called DRD4, which is again involved with regulating the size of the prefrontal cortex, which is smaller in ADHD brains. There's a DRD5 gene, which is involved with the dopamine 5 receptor, and that receptor is particularly involved in working memory, and so that could make working memory harder to function in some individuals. And then there's a general gene called SNAP25 that's involved with being able to release dopamine from one neuron into the synapse. Again, so here we have 5 different genes, all of which in some way involve the functionality of dopamine or the size of brain areas associated with ADHD. And there are differences in these genes in ADHD clients compared to controls. Finally, there are 2 genes that are involved with serotonin that we also see disruptions in ADHD, even though we don't see really disruptions in the serotonin system so much, but both of these genes, one is a receptor and one is the transporter protein for serotonin. And we think, though, both of those somehow interact with the dopaminergic system there. All right, so putting it all together, we have these genes that I just talked about, right? Those genes are influencing 2 neurotransmitters and at least 2 areas of the brain that we know are involved with ADHD. Serotonin may play a role over here, and then we have the cerebellum, which we don't have as much direct sort of genetic knowledge about how that might be involved. And then we have some basic processes that these areas of the brain are supposed to be doing. Motivation, delay aversion, reinforcement in the basal ganglia and the reward system. Executive function, behavioral inhibition, working memory in the prefrontal cortex. And then those then sort of lead to these 2 collections of symptoms. The inattentive, primarily coming out of the prefrontal cortex, and the hyperactive impulsive, primarily coming out of the basal ganglia and the reward systems. All right, when we come back, we'll spend our last hour talking about pharmacological treatment of ADHD. So let's take, again, like a 7-minute break. We'll come back at 7 after, and I will see you then. All right, so we're gonna get started back. And like I said, in this last section, hopefully you've built sort of a knowledge base of the different brain systems that we think are involved in ADHD and the neurochemicals that are involved. And so now, from a neuroscience perspective, I want to talk about the treatment of these disorders. I mean, of the pharmacological treatment of ADHD. And I know that as counselors, you all aren't prescribing ADHD meds, but I think where a role that you can really play in this whole treatment package is neuroeducation. So being educated yourself and being able to educate clients on, this is a brain disorder and there's nothing wrong with that. There should not be stigma associated with that, right? And that there are ways to treat it, and know what your options are and what the benefits and some of the disadvantages of those could be, but really sort of helping reduce the stigma that has sort of become associated with a lot of ADHD medicines. And so to start off this last segment, there is a young woman named Jessica who has ADHD that runs a great video blog it's called How to ADHD. And she has a nice little segment on the stigma that she's encountered, and actually we can hear a perspective from a doctor. So I'm gonna start off with this clip and then we'll get into some of the pharmacology of this.
Hello, brains. October is ADHD Awareness Month, and I thought it would be a good time to talk about why stimulant medication is typically used to treat ADHD, and hopefully break down some of the stigma surrounding it. Stimulant medication is considered a first-line treatment for ADHD. But when people use this medication to treat their ADHD, they often feel an incredible amount of shame. It was weird for me to even take my medication on camera, and it was probably uncomfortable to watch it. Why? Stigma. According to Cambridge Dictionary, stigma is a strong feeling of disapproval that most people in a society have about something, especially when this is unfair. And there is an intense stigma against stimulant medication. In the media and on social media, we're bombarded with misleading and shame-inducing messages.
When we think about methamphetamine, think about Adderall. Adderall is the attention deficit disorder drug that a lot of college students take. Same drug. Nobody's talking about.
It's not the same drug.
It's the exact same drug.
To it's the same thing as cocaine.
They can't eat, they can't sleep, and they're not getting better.
Isn't that in itself a symptom of taking all this essentially legal speed?
To it's a replacement for discipline or parenting.
Somebody could benefit from a little discipline.
Oh,
Don't worry, I'm not suggesting actual parenting. I'm talking about medication.
to it turns people into zombies. To it's a last resort, something to try only after trying everything else. We get these messages from documentaries, social media posts, TV shows, movies, family and friends. Some of these messages are intentionally sensationalized to get more views or shares or create moral panic.
There are cardiovascular risks, psychotic episode. You wouldn't wish that on your worst enemy.
What's the cost of that? And is that a cost we're willing to live with?
Some are meant to sell us alternatives.
Today, I'm going to share with you in this video the top foods, supplements, natural treatments, and essential oils in the treatment of ADHD.
Some are just meant to make us laugh.
And you guys currently on Adderall and want to make $50?
And some are genuinely meant to be helpful. I once had a total stranger walk up to me in a store and tell me how he managed his ADHD without medication, and I could too, as if that's how you win at ADHD, by not having to treat it. We internalize these messages, and the stigma isn't just about the medication. It's also a downplaying or doubting of the condition itself.
ADD and ADHD are not real things.
But it is ADHD.
It's impossible.
Are they anemic, and do they just need iron? That would do it. Are they just bored? Are they overstimulated with their electronic toys?
ADHD is often portrayed as a moral or parental failing, not that big a deal, or as if it doesn't even exist. In other words, not deserving of treatment. Given the sheer amount of research that exists on ADHD at this point, I'd say that's pretty unfair. It makes sense to be cautious about treatment decisions for any condition. The problem is that the stigma against stimulant medications is so strong that people end up making treatment decisions based on stigma rather than what the optimal treatment for them or their child would actually be. There's even been research done on this. Parents will often try less effective ways of helping their children with ADHD because of the stigma against stimulants. I asked ADHD Twitter to share their own experience with stigma. Their own pharmacist said taking their medication was bad for them. They've had to hide them. One person's dad used to make them hide the pills around other people. One person's mom avoided getting her diagnosed at all because they didn't want her to become an addict. Even though my mom did choose to treat my ADHD, my dad was opposed to it, and it created a lot of tension between them. I had a boyfriend once who warned me about the side effects of stimulant medication. Even though mine were really mild, he made me feel like my ADHD wasn't that big a deal. So I went off of my meds. And over the course of the next 2 years, I discovered my ADHD very much is a big deal. And there are more serious side effects to not treating my ADHD. But everyone is different. My personal experience is my personal experience. And I'm not a doctor, but Dr. Caroline Lynch Purcell's is. She also has ADHD herself and is a pediatrician who treats ADHD. So I asked her to come share what she shares with her patients about stimulant medication.
Hi, everyone. As Jessica said, I'm Dr. Carolyn Lynch Parcells. I'm a board certified pediatrician here with Girls Women and Young Men's Health and Wellness in Fort Worth, Texas. So let's address some of the misconceptions, fears, and shame surrounding medication. I'm well aware of these, not only as a physician, but because, to be honest, I had some of these myself when I was first diagnosed. I refused medication initially. A lot of us feel like we should be able to function without medication, learn all of our skills and use all of our skills and do the behavioral interventions and all of that first before we turn to medication. And in some circumstances, that may be the right choice, but that's not always the right choice. And whether it's for ourselves or whether it's for our children, medication can be a critical piece of the treatment plan. So why do we use medication as part of the treatment plan for ADHD? Well, we're still learning about ADHD, but from what we know so far, we know that dopamine and norepinephrine, two of the neurotransmitters in our brain, play a pretty large role in the challenges we face with ADHD. And while pills don't build skills, and we still need to be building those skills, skills don't change the dopamine in our brain. And the way our brains work, the difficulty we have with impulsivity, inattention, focus, make it not only a little more difficult to learn those skills, but it makes it a lot more difficult to access those skills when we need to. So the medication helps increase the dopamine levels in our brain so that our brain can function better, our neurons can communicate with each other better, we can decrease the impulsivity, decrease the inattention, focus, stay on task better, so that we can better access and utilize those skills. There are gifts to the way our brains work. We see details that other people don't see. We see solutions to problems other people don't see. It shouldn't take those gifts away. It shouldn't change who you are. It shouldn't change your personality. It should help you to have control over those gifts so that you can utilize when you use them and when you don't. One of the other misconceptions I hear a lot is that these medicines are addictive or habit forming. To be clear, when stimulant medications are used properly and under the guidance of a medical professional, the risk of addiction and habit formation is very low. On the other hand, people with ADHD do have a higher risk of addiction. The research shows us that people with ADHD, whose ADHD goes untreated or suboptimally treated, are at significantly higher risk for drug use, drug abuse, and drug dependence. What I see in my own clinic is young people coming to me vaping nicotine, smoking weed, or vaping THC in particular, to self-medicate for their ADHD, but also to self-medicate for the depression and the anxiety that is developed from their ADHD, either going untreated or not being ideally treated. And I've got to tell you, trying to help them, trying to get them off of those substances so that we can best manage their ADHD is really hard. We now have reams of data and research that show us that optimal treatment of ADHD includes medication, and medication along with behavioral interventions, coaching, accommodations in school leads to improved performance at work, relationships, decreased accidents, decreased rates of depression, anxiety, and other mental health issues. And now we even have research that shows that optimal treatment of our ADHD improves our physical health, not just our mental health. Treating our ADHD helps us to then take better care of ourselves, keep up with our doctor's appointments, take our medicines, do those things that keep us healthy. Of course, we want what's best for our kids. There's the fear of, if I do give them medicine, is it going to harm them in the long run? Is it the right choice for them? Am I making that choice for them, or am I making that choice for me? Is it because I can't handle it? Or is it because I'm not a good enough parent? If I could just parent them better, because let's be honest, that's what we hear. The other thing that I think a lot of people don't understand about ADHD is parenting a child with ADHD is different than parenting a neurotypical child. But there's this sense that we shouldn't need medication, or that medication is failure. There are always going to be those voices that judge whatever decisions we make. Those voices aren't going away, but it is not failure. Asking for what you need, asking for what your child needs, advocating for yourself or your child or your client or your patient is never failure. It's taking care of ourselves, and it's taking care of them. And you should never be ashamed for that.
I personally believe that people should do what makes sense for them and their situation. A lot of people in the ADHD community don't use medication. Some take non-stimulant medication, but before making treatment decisions for yourself or others, or adding to the stigma, I encourage you to educate yourself on what ADHD treatment actually entails. I'm grateful that my mom made the decision to put me on stimulant medication. It made a huge difference. And I'm grateful to Dr. Caroline Lynch Parcells for coming to talk with everyone.
All right. So what did you learn from that video segment? Maybe some people can unmute and tell me something that you learned from that. You knew everything before? Yes.
I did know this, but I wasn't, so I myself have ADHD. I wasn't diagnosed until I was 25, but it was not, there was no confusion. Everyone who talked to me for more than 20 minutes knew. But I remember, I I never had any big accidents, but I did always have like bumps and bruises and cuts and stuff just from what I was told I was quote unquote clumsy, and thinking that I just kind of wasn't very smart when I was growing up. And that's why I just could never get above a whatever in this class or that class, even though I tried so hard. But it wasn't until I was older, and then I remember, and then hearing it again here, that people are more likely to medicate with like illegal substances or harmful substances to cope with a lot of the problematic symptoms that either stem from ADHD or tend to accompany it, like anxiety or depression, because you can't get anything done. You think there's something wrong with you. And especially like accident stuff. Like I didn't know it, but until the statistics when I saw today that we're more likely to get into a car accident because you're somewhere else for 20 minutes and then you have rear-ended someone or something, you know, like that's pretty sobering. So that was cool.
Yeah. So, I mean, what were some of that? Those are great points. And what are particularly the risk of addiction, untreated ADHD, the impulsivity to experiment with drugs, to self-medicate, can definitely lead to higher rates of addiction in untreated ADHD. But there is research that shows that optimally treated ADHD actually has a decreased risk of substance abuse. What were some other misinformation or things that you might have gleaned from watching that video?
I think that Katie's comment in the chat is that very typical growing up in the South, spanking is very common, but you cannot spank out ADHD. I think that that's something at least we see a lot just because we work with so many small children too. I think that piece of educating the parent and even about the drug use piece of these are the things that you're looking at, not if you don't put your kid who's ADHD on medication that they're going to become addicted to drugs, but to be able to at least have that information of helping them to see the whole picture, not just I'm afraid my kid's going to get addicted to ADHD medicine.
Yeah.
The big parenting component to me that comes out in that.
Yeah. So, you know, definitely when we're dealing with a developmental disorder like ADHD, these children aren't able to make decisions for themselves or advocate for themselves. And so you're reliant on working with parents to try and make the best decisions that are going to benefit that child, right? And this misinformation or misconception that you're not parenting, it's a parenting style issue and that they would behave differently if you spanked them or if they had different means of reinforcement or punishment, all that sort of comes out, right? Some other things that I heard in the video was comparing the stimulant medication to illegal drugs. You know, the insistence, oh, it is amphetamines, and we'll talk about, or cocaine, we'll talk about why that is. And people trying to profit from fake treatments. And so a lot of the neuroeducation is for you all, when you interact with parents and ADHD, to have the necessary background that you can say, hey, I don't think taking those nutritional supplements has a research basis to, you know, these essential oils are changing dopamine levels in your brain or helping you improve, right? I think another big point is the fact that while I like the statement, pills don't build skills, and you need to build the skills, but sometimes you need the pills in order to build the skills, right? That you need to stabilize the brain in a way in order to learn effective time management. And ultimately, and I'll talk more about this, but I mean, ultimately the goal is to minimize the medication and to maximize the skill sets and the engaging of those areas of the brain so that you can minimize the drugs, right? And we've already talked about how ADHD can cause anxiety or depression, and that can lead to self-medicating and addiction, right? So understanding what are these medications to be able to advocate for your clients. There are 29 FDA approved stimulant medications, which seems like a lot. but all of those have one of two chemicals in them, methylphenidate or dextroamphetamine. So there's really only two medications, two actual drugs that are in either one is in each of those 29 approved medications. So what's the difference in all of those medications, besides whether it's methylphenidate or dextroamphetamine, is largely how it's released in the body, sort of the length or duration of the effects, and when you take those medications. That's largely the differences between them. And this is the first line treatment. It has improved symptoms in 70 to 80% of ADHD clients that take it. And it works within 45 to 60 minutes. So again, going back to this chemical imbalance theory, as soon as that drug starts improving dopamine or access to dopamine in the brain, the symptoms start to improve. The second line treatment for that 20 to 30% that don't respond to stimulant medications are what we call non-stimulant medications. And that's a little bit of a misnomer because all of these drugs are acting on norepinephrine, and norepinephrine definitely is a stimulating neurotransmitter in the brain. These may take some time to work, and what that tells us as neuroscientists is that it may not be a simple chemical imbalance, that it may be a circuit in the brain that is strengthened by norepinephrine, and it takes sustained presence of norepinephrine for that circuit to gain its functionality and start to reduce the symptoms. And then finally, unfortunately, I think, many doctors will do what's called off-label medications, and they'll take a medication that's been FDA approved for one treatment, like Wellbutrin as an antidepressant, and they'll use it for a different condition like ADHD. And, sometimes this might be effective if someone doesn't respond to either the stimulant or non-stimulant medications that are approved. Maybe this is something that could be tried. We did mention there is this relationship between serotonin and dopamine, but I think in general, understanding that off-label use is probably not the best way to medicate. A couple of things that you should know about these medications that most people probably don't know is that the effective dose is really individualistic. And the only way to know what an effective dose of ADHD medications is through trial and error, to try a particular dose, and if you get too many side effects, to then have a doctor that's responsive and will back off on that dose, or if you're not getting the appropriate reduction in symptoms, to be able to increase that dose. And for instance, Adderall comes in 7 different doses, ranging from 5 milligrams to 30 milligrams, which is a huge range, and everything in between, right? So the things that we know about that is that it's not related to sex, the dose you need. It's not related to body mass or body size. It's not related to age. And it's not even related to the severity of symptoms. Some people that have pretty severe ADHD may be effectively treated by a relatively low dose, while someone else may need a higher dose. So what drives that is that everybody's physiology is different, and how those medications affect those components of your brain are different. And some people need more of an effect to get the reduction, other people need less. All right, so looking at stimulant medications, methylphenidate and dextroamphetamine are the two classes of drugs. Two of the most popular methylphenidate drugs are Concerta and Ritalin. Ritalin was the original drug that was prescribed. These are selective dopamine agonists, meaning they increase the effectiveness of dopamine, and they do that in 2 different ways. They cause neurons to release more dopamine at the synapse, and they block that reuptake mechanism so that when dopamine is released, it stays in the synapse longer. right? Methylphenidates, well, this is Ritalin in particular, has a relatively short half-life, meaning that the duration of the behavioral effects sort of mimics that. So as the half-life means as it's being metabolized and excreted from the body. And so it has a short half-life of 3 to 4 hours. The behavioral onset, it takes about an hour to start having its effect. Some of those new 29 versions of drugs are methylphenidates that are extended release methylphenidate drugs. So it stretches that 3 to 4 hour effect out to 7 or 8 hours. And one of the disadvantages of Ritalin when it was first prescribed was that a student going to school had to stop halfway through the day and take another pill, go to the nurse, do all of that because it only lasted half of the school day. So that led to the development of dextroamphetamine drugs, and two of the most popular ones of those are dexedrine or Adderall. Adderall is probably the most commonly prescribed one. And these are what we call a non-selective dopamine agonist, meaning that it blocks both dopamine and norepinephrine. So it does have somewhat of an effect on norepinephrine as well. And it doesn't cause more dopamine to be excreted. So there's some, fundamental differences between the methylphenidate and the dextroamphetamine, even though they're both acting on dopamine. The half-life for Adderall is 8 hours, so you get that 6 to 8 hour benefit. So it's usually a 1 dose in the morning sort of drug for a child at school, which is one of its sort of big benefits there. All right, so comparing these stimulant medications to illicit stimulants, where is this misinformation coming from? Well, there is some basis for this, right? So if we look at cocaine and we look at Ritalin, methylphenidate, they share some chemical structures, but Ritalin has been modified in a way to not be nearly as intense and to have a slower onset and a longer duration. Cocaine and methylphenidate both do the same thing. So cocaine stimulates release of dopamine and blocks reuptake, which is why people say, well, that's what Ritalin does. Therefore, Ritalin's just like a watered down version of cocaine. And that's really not true. There's some other fundamental differences. The ability of cocaine to stimulate the pleasure center is much greater than Ritalin to stimulate the nucleus accumbens. Dextroamphetamine, so here's this chemical structure for amphetamine and methamphetamine. And dextroamphetamine, or Adderall, sorry, is actually a combination of 4 different dextroamphetamine or amphetamine modified molecules. So it has this thing called dextroamphetamine saccharide, amphetamine aspartate, amphetamine sulfate, and dextroamphetamine sulfate. And essentially they have taken the amphetamine molecule, so it started as the base, and they've modified it in ways that make its duration shorter and its intensity much less, and its effects in the brain much milder, right? And so it does not, again, act on those reward centers that these addictive drugs act on. It doesn't produce the highs, the pleasurable highs that these drugs produce. But that's sort of where this stigma or misinformation comes from. And so being aware of that and being able to say, yes, but it acts in a similar way, but in a fundamentally different way as well. Here, if we look at the release of dopamine in response to cocaine and in response to oral methylphenidate, what we see is that cocaine produces this intense high within 5 minutes, spiking, and it wears off within 10 minutes. Whereas we have this slow hour onset to sort of a constant level, which is what we kind of want. Remember, we want that tonic dopamine to be bathing the frontal lobes at a sort of constant lower level than happens, right? So some fundamental differences. And if we look at, I'm just gonna pull all this up here. Oops, there we go. So if we look at Ritalin compared to cocaine and we look at the elimination, why is cocaine addictive? Because it has an intense, pleasurable effect that happens very quickly within 1 to 3 minutes of being snorted or 10 to 15 seconds of being smoked or injected. That prompts, and its effectiveness is only for 15 to 30 minutes, which prompts users to use it again to get that intense high. Whereas you don't have the same pattern of use or abuse with Ritalin, right? You're not using it for the same pleasurable reasons, and you're not having to redose with it frequently. Same thing with amphetamine, very much more intense effect that's lasting for much longer in this case than Adderall. All right, so one of my questions then is, so what then makes a drug addictive? What are the qualities that make a drug addictive? And the first one is that it produces this intense pleasurable reward signal, which both Adderall and Ritalin, other stimulant medications, do not do, right? There is not this, there is a positive reward in terms of alleviation of the negative symptoms of ADHD. But that doesn't act on the reward pathway in a way that changes that circuitry, like cocaine or amphetamine does with repeated use, that shapes the reward circuitry to have to need the drug to be there in order to have proper activation. Tolerance is another thing that makes the drug addictive. That means that you need more of the drug to achieve the same effect, and that drives continued drug use and more drug use. Now, 60% of stimulant medication users do develop tolerance at some point. And in some people, the tolerance becomes such that the drugs become ineffective. But that tolerance doesn't develop in a way that taking more of the drug will give you the benefit, right? The tolerance is sort of absolute. And I'll say in that case, sometimes people have to switch medications or find that switching medications between dextroamphetamine or methylphenidate, because they have slightly different mechanisms of action, could help with that tolerance. Also, monitoring or having some off periods of the stimulant medication can help not develop that tolerance, right? When you don't need the drug, not taking it. And typically, people aren't taking these drugs 24 hours a day. They're taking them for school or for work and then not taking them at night, that sort of thing. Withdrawal is another reason that drugs are addictive. Neither of these drugs have withdrawal symptoms. Yes, the ADHD symptoms will return sometimes or often when they're not on the drug, but they don't get negative punishing effects. The body doesn't change in a way that it does with addictive drugs, so that you go through withdrawal. And then finally, almost all drugs of addiction, but particularly stimulant drugs, cocaine and amphetamines, actually impair frontal lobe signaling, whereas stimulant medications are actually improving it, right? So stimulant medications are improving decision-making and judgment, whereas these illicit drugs are decreasing the effectiveness of the frontal lobe, causing impaired judgment and decision-making. So just, some neuroeducation to help talk to people that are unaware of the difference between addictive illicit drugs and non-addictive stimulant medications. The non-stimulant medications, again, I said it's sort of a misnomer because they are acting on norepinephrine. So Strattera and Quellbri both inhibit the reuptake of norepinephrine in a similar way that we saw with methylphenidate and dopamine. This is going to increase the presence of norepinephrine in those synapses. And the other three drugs actually act to directly stimulate those alpha-2A receptors. So it acts directly on norepinephrine. All right, so what does the research tell us about ADHD treatments? And I mentioned this at the onset, but surprisingly not a lot. What we have a lot of research on is these stimulant medications, that they're effective in reducing symptoms, that they're non-habit forming, non-addictive. But that's, and that they're essentially can be as effective as behavioral therapy as well. But beyond that, we haven't done a whole lot of research on these. It's sort of been about the efficacy of them and not really on long-term effects, or actually how are they doing the improvements that they're doing in the brain. And part of that is that ADD was the original diagnosis in 1980, and ADHD became a diagnosis in 1987, which really wasn't that long ago. So this hasn't been like a diagnosable disease for that long, that we've been able to accumulate that much research on it. And the FDA didn't approve drugs until 2002, was the first, was when Ritalin was first approved, and Adderall a couple months later. So in terms of being able to accumulate research on long-term effects of these drugs, we don't really have a lot on there. Another difficulty in doing research is it's really difficult to create an animal model of ADHD. So we can give ADHD drugs to rats that don't have ADHD, but then you're giving an animal or a person a medication when they don't have a deficit in dopamine. So you're actually pushing them off of the top of that U-shaped curve towards the too much dopamine area, right? Which is why it is a big problem when students that don't have ADHD are buying ADHD meds and taking them as stimulants, because they can act as a stimulant if you have enough dopamine. If you're not replacing missing dopamine, right, you're getting excess dopamine. And that's also where some of this sort of addictiveness has come from, because some people who do abuse ADHD medications, but they don't have ADHD. And most of the human research to date has been done in adults and not in children. And so it's much more difficult to do research on children, but we need to, and particularly because we don't really know the effect of the continued presence of these drugs in a developing brain, and how that might shape its development, right? And so that's research that's kind of missing that we need to know. Some of the stuff we do know, this is a study that looked at CBT treatment and CBT treatment plus medication, and they saw the same benefit of CBT treatment and they didn't see a further improvement of that CBT treatment when they were given the medication. So essentially, learning skills, learning how to cognitively control brain activity, internally control your cognition through CBT can reduce the severity of ADHD. And you may, as I mentioned before, need the pills in order to get the brain in a place that you can employ CBT effectively. But the goal would be to wean the clients off of the medication at some point, or at least maybe even reduce the medication levels, or reduce how often they're taking it, so that when they're off their meds on the weekend, they can effectively time manage, they can get their chores done, they can cope, they can do all the skills that they need to do off medication. One of the things that's differences between CBT and medication is when the pills wear off, the symptoms come right back. Right? With CBT, you now have a tool to deal with those symptoms, right? So relapse in people that have undergone behavioral therapy or coaching therapy is much lower, right? They're much more functional off the meds than people that simply rely on the medications to improve the symptoms. There have been a few studies that have looked at sort of long-term effects of being on these medications. And so this is a study that looked at the expression of GABA. If you remember, GABA is the main inhibitory neurotransmitter, sort of used to turn neurons off in the brain. It works with with glutamate to do the on-off signaling. And in this case, in this study, they had 3 different groups. They had a group, the STN or open bar group was a control group that had no exposure to Ritalin, methylphenidate. EST group is people that were put on Ritalin before they were 16 years old, and they continued on Ritalin. So Ritalin during brain development, or that final adolescent maturation that was occurring during the teenage years. And then people that were put on Ritalin, but after they were 23 years of age. So brain development was pretty much done by that time. And so, to explain this, if we look here at this graph that says before MPH, what they did is both of these groups were being prescribed Ritalin, and they took them off Ritalin for 5 days, and they gave them what we call a washout period to get it out of their system. And so this is the amount of GABA that's present in the brain without being on the medication. And if we compare to controls, the people that were put on Ritalin after development don't show a difference. However, there's a decrease in the expression of GABA in the brains of individuals that were on Ritalin during that final stage of brain development. And then they gave them a dose of Ritalin, of methylphenidate. And so here, after being given the dose, those decreases in GABA came back up to the appropriate level, right? That we see in controls. And so this is acute, taking a pill of Ritalin increased the GABA. So things that this doesn't tell us. We can imply, hey, the fact that they were on Ritalin while the brain was still developing caused this decrease in GABA. No, we can't say that for sure. Maybe this decrease in GABA was what was causing them to need the Ritalin, and it wasn't that the Ritalin induced it. Maybe it was present before as part of the symptoms before that. We don't know the answer to that, right? We can say that the Ritalin helps stabilize the brain activity so that GABA's at the normal sort of levels. And it doesn't look like we see a similar decrease in GABA for people post-development that started taking it long-term. So, this warrants being cautious with stimulant medications during development, taking off periods, summertime, times at night, the weekend, times when clients can employ behavioral skills instead of taking the medication to be successful in what they're doing. I mentioned that the brain compensates for the continued presence and that tolerance can develop in these stimulant medications. This is a study that measured the amount of dopamine reuptake transporters. So that's the thing that that Ritalin and Adderall are blocking in order to induce more dopamine in the synapse. And so this is a ADHD patient on medication, ADHD control patient. And 1 year after being on Ritalin, what we see is the patient was on Ritalin, much darker, higher levels of dopamine transporter expression. So this means in the 12 months that that person was on Ritalin, the brain said, hey, you're blocking my ability to recycle dopamine. I'm gonna put, instead of having 5 of those transporters out there, I'm gonna put 15 of them so that I can recapture so I can compete against this drug effect and recycle the dopamine, right? So what does that mean? It means that in this person, likely after 12 months, that drug has lost some of its effectiveness because the brain is compensated by not allowing it to block the dopamine as much by being able to recycle more, right? The second thing that means is that when this person now stops using Ritalin, instead of recycling 5 molecules, it's now recycling 15. So it's actually going to pull dopamine out of the synapse much faster than it should be doing that, right? So that sort of could mean that the symptoms might be exacerbated. You would actually be decreasing dopamine when you're not taking the drug. So again, these are things that we have to be careful with. We need to make sure people are taking these medications in ways that tolerance isn't occurring, right? All right, so finally, we'll sort of wrap things up here, and I'll check that, the chat real quick. I see, yeah, the intuitive is acting on the norepinephrine reuptake inhibitors. It's a newer class of drugs, which they call non-stimulant. So what can counselors do in the role of treating ADHD in light of what we've sort of talked about today? Certainly the comorbid things that are occurring, mental health issues. There's a need for treating those and helping people become neuroeducated and reducing the stigma on diseases of the brain, which this really is a different brain state, right? In terms of tools for some of the emotional regulation, so we've talked a lot about the inattention part, but some of the emotional impulsiveness. One of the things that works really well, we talked about this, I think, when we talked about anxiety, is an abbreviation TIPS that stands for temperature, intense exercise, paced breathing, impaired relaxation. So when that emotional, not being able to regulate those emotions starts to arise, by taking a wet washcloth or an ice pack and putting that on your arm or your head, that can actually help reduce those emotions. Jumping jacks or push-ups, some kind of intense exercise can alleviate that. Going on a stiff walk, right? Paced breathing, so and paired relaxation, but two relaxation techniques. Paired relaxation is where you tense that muscle group and then intentionally relax it. So you're pairing contraction with relaxation there. And then I think I'm not telling you all anything new. This is what you know and do, right? Is behavioral classroom modification practices. A lot of times those involve token reward types of systems. And what do we say about how ADHD affects the reward systems in the brain? Does anyone want to chime in and say, if they remember what we said, how they're modified from a neurotypical brain to an ADHD brain?
Was it that it was more difficult to pair the cues with the reward?
Yeah, and in particular, ADHD brains favor smaller, more frequent rewards than larger, more delayed rewards. So in developing these classroom practices, the best types would be ones in which you could deliver small, immediate rewards so that they can start to begin some of that cueing behavior and hopefully link those cues together, right? Organizational training, and there's a lot of individualistic tricks and tips. And so everybody's ADHD gets expressed in different ways, and some people may be more of the type that they lose things more frequently. And so there's a whole ADHD online supportive community that are like, buy a bigger phone. The bigger phone you have, the harder it is to misplace it. Have a big keychain. I mean, one of the, if time management is a problem, having the presence of clocks and wearing a watch is one way to bring that to your awareness, right? So there's lots of tips that can help with this. One of the funny things that I learned from the Holderness family from Penn is that he often would put things on top of his car and then drive off with them on top of his car. So he now has a magnet on his car, on top of his car, that he sees, and it reminds him, hey, check up here, make sure there's nothing up here before you drive off. So depending on what it is for individual things, you can you can tailor that by listening to clients and hearing what it is that they need. the last thing I'll point out that I found with my research on this is what about educational accommodations? And I know the doctor in that video clip mentioned these, but the research actually shows that that educational accommodations are often not very helpful with ADHD. In fact, the most common sort of accommodations are having extended time on tests. And often, sometimes ADHD clients will say that actually exacerbates their ADHD because they have more time to wander and less reason to do time management and planning. And so we have to be careful about the role of that. In general, the types of accommodations that are best helpful for ADHD are what we call universal accommodations, which are things that help anybody do the test better by having more organized tests or so things that can help anybody learn better are what's going to help people with ADHD learn better as well. All right, that about wraps up my time. Do you all have questions for me? Did I get everything in the chat? Yes, having one M&M per paragraph rather than a bag at the end of the chapter. Great use of smaller immediate rewards. Any other questions?
All right. Any other questions? If not, guys, if you will go ahead and type your name into the comments as you're signing out. There will be the PowerPoint will come with this whenever you fill out your evaluation, which was in the link. And the link was in the email that I sent. If somebody will grab that link for me, please, and throw it into the chat. And I want to make sure that it works for everyone. Sometimes I forget to hit a setting and people who don't have a GCC email have a hard time getting it. So
Dr. Pittman, did you have any information about the ADHD question of males versus females?
About why we see it more in males than females?
It was in the chat as far as because you were talking about how the males were more diagnosed than the females, but I just wasn't sure if there was more research.
Yes, so that's actually that's a good point. And yes, there has been way more research on males than females, and that's a recruiting issue, right? So it's just, there's more availability of male subjects than female subjects, but because there is a pretty big sex difference, that's something we really need to explore and try and understand that interaction. So.
All right, guys. I appreciate this. I will get your CEU sent out. We asked for 2 weeks, but usually I'll try to get them to you in the next couple of days.